Jonathan Brestoff, who will be talking about mitochondria transfer-based therapies, reduce the morbidity and mortality of Leigh syndrome. Dr. Brestoff, whenever you are ready.
Thank you so much. First of all, I want to thank the organizers for finding some space in the agenda to include this presentation and to share our work with you. I'm really excited to share a new paper that we just put out in Nature Metabolism a couple of weeks ago. That paper is about mitochondria transfer-based therapies for Leigh syndrome. These are my disclosures. The most relevant ones are that my lab receives research support from Luca Science for an unrelated project, and I'm also on the scientific advisory board for that company. So mitochondria arose from an ancient endosymbiotic process and are now the energetic hubs for essentially every eukaryotic cell type. It's thought that these mitochondria arose from endocytosis of a proteobacterial species that underwent an endosymbiotic process over 1.6 billion years ago to give rise to what we know of as mitochondria today. There are a number of relics of their bacterial ancestry, including the presence of a circular double-stranded DNA genome that's about 16 kb in vertebrate animal species. And these mitochondria are, of course, most famous for their role in energy metabolism, but they have many other important functions, including serving as signaling centers and regulation of nuclear gene expression, cell death, and cell cycling. The textbook thinking about mitochondria propagation between cells is known as vertical inheritance of mitochondria, where the parent cell divides the mitochondria symmetrically or asymmetrically to the two daughter cells. But in the last 20 years or so, we've started to appreciate that many cell types actually possess the ability to eject their mitochondria and transfer them to developmentally unrelated cell types in a process that's been termed horizontal or intercellular mitochondria transfer. This process appears to be deeply evolutionarily conserved and has been identified in diverse organisms ranging from yeast, plants, fungi, mollusks even can transfer some of their mitochondria to other cell types, fish, and vertebrate animals, including mice. It's also been identified in human cells in vitro, but not in humans in vivo yet. Mitochondria transfer has been identified in many different organ systems and has been linked to both physiologic and pathophysiologic states. Many of these we recently discussed in a recent review article in Nature on this topic. And just a couple of the ones that I think are most relevant for this particular talk are that it's been shown that astrocytes actually can transfer some of their mitochondria to neurons in the setting of ischemic stroke to support neuronal survival, and that this is important for regulating stroke outcomes. In addition, cancer cells actually hijack this process and they steal mitochondria from tumor-associated immune cells like tumor-infiltrating lymphocytes or TILs. This simultaneously impairs anti-tumor immunity, but also provides energetic support to these highly energetically demanding cancer cells. My lab is primarily focused on studying metabolic disease pathogenesis in the context of obesity, and so we're really interested in the adipose tissue and wanted to visualize this process unfolding within that organ. So to address that, we performed this experiment where we isolated wild-type macrophages from white adipose tissue and labeled them with CM-TMR, which is a cytotracker dye. And then we adoptively transferred them into global mitochondria reporter mice, which are labeled as MTD2. And then the next day, we performed intravital two-photon microscopy of the white adipose tissue to identify the adoptively transferred immune cells in the tissue, where we can do intravital two-photon microscopy for 3D reconstructions. Here you can see a red donor macrophage that's intercalated among a number of adipocytes, which are these enormous cells with a large lipid droplet. And then there are the green puncta, our host mitochondria. In every example that we could find, we observed that there was uptake of donor-derived mitochondria into the macrophage. And on an analysis, we even found evidence of a mitochondria capture event where a green puncta is internalized into the cell. And you can even see it do fusion and fission with other mitochondria. Here we're moving the field of view through a still image and then through a moving cell, just so that you can see that these mitochondria are really in the cell, not sitting above or below. So I think these are actually the first visual demonstration of mitochondria being transferred from one cell to another in situ in a living tissue. In a number of papers, we've done work that establishes that under healthy conditions, adipocytes actually frequently transfer their mitochondria preferentially to tissue-resident macrophages, and that this process actually becomes impaired in obesity due to the presence of dietary long-chain fatty acids, which suppress the ability for these macrophages to take up mitochondria. And this actually diverts the adipocyte-derived mitochondria into the bloodstream and allows these mitochondria to be redistributed to other organs, including cells in the heart, likely endothelial cells. And this process, through work from Clara Crew, has shown that that this process actually elicits an antioxidant gene expression response that protects mice from subsequent ischemia reperfusion injury, suggesting that this interorgan mitochondria transfer has the capability of metabolically preconditioning cells. Question that we are really interested in is whether or not mitochondria transfer could actually contribute to the cellular metabolism of macrophages or any other acceptor cell. And I really like to use genetic models, and this is what introduced my laboratory to Leigh syndrome and NDUS4 mutations. We thought that maybe we could actually rescue cellular metabolism by using purified mitochondria to rescue cell intrinsic defects in metabolism in cells that lack complex 1. And work that was led by Rocky Giwa and Wentong Jia in the lab, they did this experiment where they isolated mitochondria from global mitochondrial reporter mice, and then this is from the liver, and then injected the mitochondria by intraperitoneal injection into either wild type or NDUS4 deficient animals. 24 hours later, we harvested the macrophages from these animals and then did seahorse mitochondria stress test assays. What you can see is that in the red open squares, the NDUS4 deficient macrophages have severely depressed aerobic oxygen consumption rates under basal and also under maximal oxygen consumption rate conditions. But one dose of mitochondria the day before completely rescues the aerobic respiration rates back to wild type levels, which is shown by the closed red squares. Interestingly, in the wild type cells, there's a really minimal, if any, impact at all, which has led us to suggest that there may be actually context-dependent utilization of these captured mitochondria, where under healthy conditions, those mitochondria are preferentially degraded. But if there is metabolic catastrophe or metabolic crisis, cells can plug into these mitochondria on a temporary basis. Of course, the NDUS4 mutation is famously associated with Leigh syndrome, which is a fatal mitochondrial disease that really has no effective FDA-approved therapies. There's been so much discussion about this disease already, so I'm not going to go into all of the details about this, but essentially, we thought that maybe we could harness this biology to treat Leigh syndrome. Our initial hypothesis was that maybe we could engraft the animals with donor-derived healthy immune cells that would allow for these cells to basically be systemically distributed, and they could donate healthy mitochondria to disease cells in the context of Leigh syndrome. To first address this question, we lethally irradiated NDUS4 deficient animals and then transplanted them with either CD45.1 wild type or CD45.1 MTD2 bone marrow, which are mitochondrial reporter mice bone marrow. Then 2 weeks later, we harvested the plasma blood from these mice and did small particle flow cytometry on the cell-free plasma and found release of these donor-derived mitochondria in the blood with a roughly 300,000 to 400,000 cell-free mitochondria per mil. We've also observed mitochondria transfer from these donor immune cells to many different cell types in a number of different organs, but that data is not shown here for sake of time. Next, we tested whether or not transplanting wild-type bone marrow could potentially ameliorate Leigh syndrome, morbidity, and mortality. And so in this experiment, we lethally irradiated wild-type or NDUS4 deficient animals and then did bone marrow transplants with either wild-type or knockout bone marrow and to generate these bone marrow chimeras. And what we found in our survival curves was that the wild-type bone marrow transplantation markedly extended the survival of these animals. You'll notice that the survival for the control group is substantially shorter in this case than what you find in the literature, but this is because the mice are lethally irradiated, and NDUF4 deficient animals are very susceptible to radiation-induced death. In fact, we use a lower dose than we would use for a wild-type animal for radiation. When we put these mice on the rotarod to assess for neurologic function, we found that the wild-type bone marrow transplantation was associated with a much better performance on the rotarod. They were able to maintain balance at faster rotational speeds and for longer. Based off of the association with mitochondria transfer from the donor immune cells to all the other cell types, many other cell types in the NDUS4 deficient animals, we hypothesized that maybe we could rescue Leigh syndrome or ameliorate Leigh syndrome through administration of isolated mitochondria in a procedure called mitochondria transplantation. In this experiment led by Stella Varnum, she administered either PBS or purified wild-type mitochondria from the livers of healthy mice to NDUS4 deficient animals and observed that true survival was significantly improved with mitochondria transplantation. We actually use true survival rather than euthanasia criteria because we feel that many of the euthanasia criteria are just single point estimates of future survival that are not really reliable. So this is actually true survival, which is a category E procedure that we have approved. This is what the animals look like at 42 days. This represents one of several of experiments. They don't always look quite like this, but this is a pretty good representative example. And you can see that there's pretty rapid regrowth of fur when alopecia occurs. And then in the PBS-treated animals, you see evidence of hind limb paralysis. There's this leg splaying. They fail to shred their nestlets well over a 24-hour period. And then we have to use hydrogel to give them some kind of hydration. Whereas the animals that had received isolated mitochondria shredded their nestlets, were more robust looking, they regrew their fur, and they were more exploratory. They're not completely normal. You can see a head tilt in this animal, and this animal actually does a circular motion, and you can see that video online. We also performed neurologic function testing on these animals and found that the mice that received mitochondria could maintain balance on a stationary rotarod much longer than the PBS-treated animals. And they also had slightly improved grip strength. In those experiments, we were using PBS as a control, but it's not the ideal control for exogenous mitochondria. So in this case, we isolated either wild type or knockout mitochondria and administered them to NDUS4 deficient animals. And also, first we confirmed that the knockout mitochondria had impaired aerobic respiration basally on a seahorse assay. And then when we administered these two different mitochondria to the NDUS4 deficient animals, we found rescue in rotarod performance with the wild type mitochondria, but not the knockout mitochondria. Next, we collaborated with another laboratory through a connection with Luca Science to use the Luca Science isolated mitochondria product called MRCQ to test whether or not this product might also ameliorate Leigh syndrome in the NDUS4 deficient animal model. MRCQ is basically it's an isolated cell-free mitochondria product that can be freeze-thawed. And so all of these data are actually post a freeze thaw. You can see that the MRCQ is able to produce ATP that is impaired with oligomycin treatment, and much better ATP production rates post a thaw compared to conventional mitochondria isolation methods. They have better outer mitochondrial membrane integrity and better inner mitochondrial membrane integrity after a freeze thaw. Then our collaborators in the Yokota lab at Osaka University administered either PBS or MRCQ to NDUS4 deficient animals and observed a similar phenotype where there's an extension of survival and also improved hair regrowth. They have longer hang wire times, suggesting improved grip strength. They have a shorter writing reflex, where shorter writing reflexes are better than longer reflexes. And this is what the animals look like when they're hung by their tail. The MRC treated mice are able to maintain balance and are stable, whereas the PBS control treated mice they gyrate unstably, suggesting an ataxic or a discoordinated movement phenotype. Alessandro Bitto published this really nice commentary on this work that is a companion piece to our recent article and summarizes this beautifully. I think that we've identified two potential mitochondria transfer-based therapies. One is through bone marrow transplantation or stem cell transplantation, and the other is through mitochondria transplantation, which appear to ameliorate Leigh syndrome in terms of both symptomatology and their survival. We're now working with Russell Senato at Seattle Children's Hospital to design an initial clinical trial to establish whether or not the mitochondria product MRCQ may have therapeutic utility to treat inherited mitochondrial diseases. Thank you so much for all of your attention. And I want to also acknowledge all of the people in the laboratory who did all this work, especially Rocky Giwa and Wentong Jia, who led the initial in vitro studies, and Stella Varnum, who led all of the in vivo studies in our laboratory on NDUFS4, and the Yokota lab with Ritsuko Nakai, who was the lead from the Yokota lab. Thank you.
Great. Thank you so much.